Hello everyone. Today I have somewhat of an odd question for you all. Have you ever seen someone drunk? Think about the way that they act, behave, and speak. Today, this is exactly what our project will be addressing. My name is Srilakshmi Nair.
My name is Vishva Patel.
My name is Kanwalpreet Kaur.
Today we'll be presenting Mission Tipsy Planaria, the effect of dandelion root on behavior recovery.
Acetaldehyde found in ethanol produces free radicals in your body. This experiment was conducted on a Planaria species, Zigzia tigrina, as they have a similar nervous system to humans. The positive behaviors that they display are the formation of tilting, heads and tails, active moving, and regular body shapes, and the negative behaviors that they display are the formation of C-shapes and screw-like shapes. Dandelion root powder is a powerful antioxidant which contains phenolic compounds and reduces oxidative stress. It was hypothesized that dandelion root powder can alleviate the negative effects of ethanol. There were 3 different groups, the during, after, and post-treatment, and there were 3 different concentrations of the solution. For trial 1, the x-axis represents the number of groups, while the y-axis represents the frequency of planaria exhibiting the behavior. For trial 2, the y-axis represents the percentage of planaria exhibiting the behavior. Now, Srilakshmi Nair will be discussing the results.
For our results, we collected data in 3 different trials, which included a during, after, and post-observatory period. C-shapes are classified as negative behaviors in planaria, and it results from disturbances within them. Trial 3, Figure 1, Panel A significantly proves that higher concentrations of dandelion root powder are optimal in reducing C-shape formations, especially during a 20-minute treatment. And a p-value less than 0.05 was derived between the control and 4% group. Additionally, an R-squared value of 0.95 was also derived, indicating further significance. It can also be seen that the control group had the highest frequency of C-shaped formations, at 4.4. Additionally, the 1% and 2% have a lower frequency than the control group. Now, Vishwa Patel will be furthering our data analysis.
Trial 1 showed an increase in the positive behavior of tilting of heads and tails. Trial 2 demonstrated a decrease in the negative behavior of screw-like shapes and an increase in the positive behavior as well. Trial 3 depicted a decrease in the C-shape formation, specifically in the 4% group. A major improvement made in our data analysis was that in trials 1 and trials 2, we looked at the number of planaria that showed the behavior and averaged that. However, in trial 3, we looked at the number of times each planaria showed the behavior and averaged that. This allowed us to utilize ANOVA. The data shows that high concentrations of this natural antioxidant is beneficial in reducing the negative behavioral effects of hangovers. This poses the question if other antioxidants can be a treatment to ethanol intoxication.
Good afternoon. Get out your brooms and dustpans because it's about to get dusty. I'm Miriam.
I'm Felicity.
And I'm Areej. Before we begin, we would like you to keep in mind that this project consists of 2 different parts that are only correlated by the construction site. Imagine walking outside to hear a large crash and see a pile of dust plummet into the air. Well, welcome to the reality of Francis Lewis students. Currently, our school is undergoing construction, and this made us wonder whether inhaling the pollution commonly caused by construction sites would prove to have a harmful effect on students and also the environment. In this experiment, street dust and C. elegans were used.
So how we tested this is we had an ecological assessment that would assess the characteristics of the trees. And then on the other hand, we had created dust solutions from the dust we collected from 3 different sites and placed them on the C. elegans, and every day we took pictures as well as using an M9 buffer to have a behavior assessment. 2 notable graphs include the tree species composition as well as the leaf health.
As Miriam mentioned before, there are 2 parts to this experiment. In the dust part, where we did C. elegans and dust, we did a total of 3 trials. Unfortunately, according to ANOVA, all 3 trials were not statistically significant. However, in our ecological assessment, we found much more interesting findings. We saw, as Miriam mentioned earlier, that the leaf health was very poor in the trees around our school. This could indicate that they are going through drought stress. And we also saw that the trees around our school, most of them are introduced. And that means that they're not from New York City. So this highlights the importance of keeping our urban ecology healthy. And in the future, we want to We want to investigate what really is causing the drought stress in our school.
This experiment is important because it is the first ecological assessment that has been done from Francis Lewis. It is also highlighting the importance of keeping our flora and our fauna safe in the wake of modern urbanization. Thank you so much.
Thank you.
Have a wonderful day.
Hi, my name is Meghan Stern. I'm a senior at Midwood High School, and I conducted my research at Brooklyn College in the Biomimetics and Cognitive Robotics Lab, under the supervision of Dr. Frank Grasso. The title of my project is Home is Where the Support Is, which is indeed true. You can't have a house without a good foundation. In my study, we found this to be a possible reason for why monk parakeets choose certain structures to build their nest on. Monk parakeets are originally from Argentina, but came to the United States in the 1960s through the pet trade. In New York, monk parakeets are found in various neighborhoods in Brooklyn and some in the Bronx. Interestingly, in a survey conducted by the lab in 2013, we found that 97% of recorded nests were found on man-made structures, while the remaining percent were found on trees. This statistic was shocking to me, considering monk parakeets' native country is much more rural than New York, so I began to further investigate. In my study, I had a sample size of 22 images, all photographed from below. Then, in Adobe Photoshop, I found the area of the nest and the area of the substrate. The substrate is the surface in which the nest was built on. I then took a ratio of the area of the nest to the area of the substrate and conducted 2 t-tests. In both t-tests, I got a p-value that was less than 0.05, indicating that my results were statistically significant. Monk parakeet nests built on man-made structures were bigger than nests built on trees. A reason for why the nest may be bigger is that the surface of man-made structures are often flat, so it gives monk parakeets a stable surface to build off of, and it's much easier for them to weave their sticks. Overall, they're allowed to be more adventurous, so this may be a key factor in why they prefer man-made structures. However, a problem comes with this. Oftentimes, when monk parakeet nests become too large, it can cause these man-made structures to result in blackouts or sometimes even fires. So it's important to find a way to ward off monk parakeets in an ethical manner.
Hello, my name is Andrei Supershow. So, iron. We all know what's iron. Iron is one of the most abundant elements on the Earth. However, this abundance sometimes can lead to troubles when iron gets accumulated in the sources of drinking water. When the iron level is too high, the water can become unsafe to drink, which is dangerous for some regions of the world where the well water is the primary source of drinking water. Therefore, knowing that plants and fungi are able to absorb heavy metals, I decided to use these organisms to clean water from iron. So for my research, I chose Oryza sativa and Hordeum vulgare, commonly known as rice and barley respectively, to clean the water from iron-3 ions. Also, I wanted to see the effect Aspergillus niger or black mold has on the absorption rate. And finally, I wanted to investigate the effect iron has on the growth rate of the plants. So to test the hypothesis, I set up several hydroponics. Some of them were filled with iron 100 ppm iron solution, and the rest were filled with regular distilled water. To test the growth rate, I was measuring the average length of the shoots of the plants in each group and recorded this in these 2 graphs. At the bottom, you can see the growth rate. At the bottom, you can see the growth rate of Hordeum vulgare that was growing in iron solution. And the same goes for Oryza. And at the top, here is the growth rate of the plants which are growing in regular water. Again, the same for Oryza vulgare and Oryza. Then, at the end of the experiment, I recorded the amount of iron absorbed in by each plant. So how can we see here the difference between amount of iron absorbed by plants with and without fungus is statistically insignificant. So it cannot be told did the fungus have any effect on the absorption rate or not. So this part, I found that fungus has almost no effect on the growth, on the absorption of iron. So then these two graphs show that plants' growth is affected a lot by the iron present in water. And the third part, that plants were actually able to decrease the amount of iron present in water. So one of my most important findings in this experiment is that plants actually began to die at 100 ppm of concentration of iron. I couldn't find, while making the research, any evidences showing the amount of iron dangerous for Hordeum vulgare and Oryza. Therefore, I had to guess, and it turned out that 100 ppm is dangerous for these plants. Therefore, I would like to conduct several more experiments with other concentrations of iron in order to find the borderline at which iron becomes dangerous to the plants. So that's pretty much all for this research. Thank you for listening.
Hello. So the inspiration for our project is primarily around tuberculosis. Now, tuberculosis is a major problem, and it's one of the leading causes of death, especially in developing nations. Tuberculosis does have an antibiotic named rifampicin. However, rifampicin-resistant tuberculosis is becoming increasingly more prevalent. Now, in an effort to try and solve this growing problem, we need to look at the biological process that is bacteria acquiring resistance. Now, enzymes in general are very efficient, but in order to develop resistance to certain antibiotics, certain bacterial enzymes need to undergo mutations. And upon acquiring mutations, they become less efficient at their original task. Now, this is formally known as a fitness cost. And as it pertains to tuberculosis, it functions very similar. So tuberculosis bacteria, upon acquiring resistance to rifampicin, also are known to exhibit hypersensitivity to compounds called fluoroanalogs. Now, fluoroanalogs are common chemotherapeutics that have been used in a treatment setting before. So the purpose specifically of our project was to identify genes which are responsible for this hypersensitivity and studying all potential gene targets involved with the processing of fluoroanalogs. Now, to do this, we specifically looked at cultures related to fluoroanalog resistance. So what we did was we isolated resistant mutants that are exhibiting resistance to fluoroanalogs in order to study the mechanism for why cultures might not be resistant. And through gene sequencing, we discovered a mutation in a key salvage pathway gene, which is strongly correlated to be the cause of this acquired resistance. And this is very significant because it highlights the potential that this gene has in playing a role in the combination treatment of rifampicin and fluoroanalogs as a solution to this growing problem of rifampicin-resistant tuberculosis. Thank you.
Hi, my name is Noah Gordon. I'm a sophomore in the Edward R. Murrow High School MSTAR program, and my project researched how Pavlovian learning mechanisms could be used to alleviate nicotine withdrawal symptoms in Yushavaiev tigrina. Addiction affects millions in the US, and around 20 million Americans have a substance use disorder. Nicotine dependence has the highest rate of any substance use disorder, and nicotine and smoking can cause lung cancer, among other issues, and lung cancer is one of the highest causes of death in the United States. Volley and teen vaping have made the issue of addiction, especially nicotine addiction, more prevalent in the recent years. And what my project studied, which was environmental cues, which are commonly known as triggers, these can be learned with as little as one exposure, which is especially important because teens are doing all sorts of things, and so is everyone, and it's best that we best understand how to prevent withdrawal and treat addiction. So for my procedure, there were 2 groups, each of 10 Dugesia tigrina. They were placed in a petri dish containing only distilled water, and they were shielded from all light. They were then switched to a petri dish containing a nicotine solution, and they were given the availability cue, which was the red light. They were cycled back and forth between these 2 environments and their respective cues. After these cycles, they were left in the nicotine solution for around 40 hours, and then with the availability cue, and then they were taken out of the nicotine and put in distilled water, effectively putting them through withdrawal. Group 1 was given the availability cue, so during withdrawal, they were given the red light, so they thought that there should have been nicotine in the environment, and Group 2 was given the emission cue, so they were shielded from all light. The control group was stayed only in distilled water, but was exposed to the cycling red light. And what I found was that there was correlation between the drug emission cue and reduced symptomatic behavior. So Group 2 showed minimal adverse side effects compared to Group 1, which showed a lot more severe side effects. And there was attraction demonstrated to the red light, which demonstrates a desire for nicotine. There was lack of exploratory behavior during initial withdrawal, and that may be indicative of decreased cognitive or locomotor functioning. I noticed when I did a modified conditioned place preference at the end of the experiment, I noticed that Group 2, which was given the emission cue, still demonstrated a very strong attraction to light, and Group 1 did not, which showed that they had partially unlearned the cue. So while their withdrawal was worse symptomatically, they had partially unlearned the cue, which has implications for how people do cue exposure therapy to treat addiction and other mental health issues. Thank you. My name is Noah McGuane. My mentors are Mr. Howie Wong, Ms. Ray Chan, and Ms. Jia Franco.
My name is Lily O'Sullivan, and I am a senior at the Bronx High School of Science. Have you ever heard of MRI scans? Probably yes. How about diffusion MRI? Probably not. Diffusion MRI is a groundbreakingly sensitive form of MRI. This sensitivity is essential for analyzing and detecting the effects of prevalent neurological diseases on the brain, because often these diseases only cause very subtle abnormalities in brain matter. Diffusion MRI is a relatively new technology, so there's not that much accessible data out there. But since diffusion MRI, otherwise known as DMRI, can provide such valuable insights to scientists have begun pooling DMRI data across the world to increase the sample size of their neuroimaging studies. With this greater sample size comes the potential for studies to map the brain, study brain-related diseases with lots of details, and even explore human cognition. However, there's a catch. When you collect data from a wide variety of sources, it can often compromise the data because different technology or procedures in different locations or studies can introduce unwanted variability in the data. Thus, to allow researchers to continue pooling DMRI data, scientists created a technique known as harmonization, an algorithmic process that adjusts originally incompatible data to make it possible to use data from a wide variety of sources in just one study. In recent years, other scientists have been developing different harmonization algorithms. These scientists have achieved great degrees of success in removing technological variation introduced by location, site protocols, and scanners. However, none have been able to analyze or overcome the variation introduced by image processing algorithms, which is where my study comes in. I analyze how image processing algorithms affect DMRI data and work to overcome the mathematical incompatibility between these image processing algorithms, which are necessary to process the raw data in every single neuroimaging study. My results were very promising. Although I originally showed how image processing algorithms can create great disparities in DMRI data, my use of the combat harmonization algorithm actually removed all the random variability that the image processing software introduced. My work helps elevate neuroimaging studies to the next level, with the ability to increase the size of datasets by pooling data from a variety of originally mathematically incompatible sources. Without compromising the validity of the data, scientists can now elevate their study of neuroanatomy and highly prevalent neurodegenerative and psychiatric diseases. I hope scientists continue to build on my work, because together we can open the door to a new world of neuroscience.
Let's start off with a question. How many hours do you think teenagers spend daily on their social media? 1 hour? 2 hours? Maybe 3? How about 9 hours a day? The various amounts of information you can stumble upon is vast when spending 9 hours daily on social media. Could this consumption of content lead to changes in a teenager's development into an adult? Could this impact their social skills, productivity, self-esteem, mental health, and body image? Hi, my name is Tara Riley, and I'm studying the impact of Instagram on adolescents' eating behaviors. My research examined the effects of the types of accounts teenagers follow on Instagram and their eating behaviors.
Although there has been research investigating social media such as Facebook and their connection to negative eating behaviors, there has been no research diving into the world of social media primarily used by teenagers nowadays, such as Instagram, Snapchat, and Twitter. Many of these previous studies have primarily been conducted on young adults, but have not looked into the effects of various social media platforms on teenagers. This is the first study to evaluate the type of accounts teenagers follow and how this can affect their eating behaviors, which can lead to various eating disorders such as anorexia nervosa, bulimia nervosa, and binge eating disorders. Although this study shows no correlation between accounts followed on Instagram and their eating behaviors, the fact that 37.5% of participants exhibited negative eating behaviors is startling. The widespread use of social media and accessibility of information and misinformation shows the need for further research in the influence social media plays in the development of eating disorders, especially in minors. Thank you.
Good morning, ma'am, sir. I hope you're doing well. My name is Jocelyn Kaur, and this is my partner, Miriam, and today we will be presenting to you our project. Our project's name is Will Survive German, One Essential Beaker at a Time, Effective Odor Stimulants on Learning Introductory Material for a New Foreign Language. Generally speaking, the main purpose of the experiment was to find out whether the presence of citrus essential oils will actually help someone learn a foreign language. Our methodology focused on learning introductory material for a foreign language through the auditory paradigm and the read-write paradigm, and there was various trials. Some trials that focused on no essential oil and one that had with essential oil. And we actually did this to see the effects of citrus essential oil in the language learning experience and process. So now Miriam will actually be explaining the conclusions and results.
So in conclusion, in both paradigms, participants perform significantly better when exposed to citrus oil, regardless of their GPA, gender, grade, and their preferred learning styles. When exposed to the same well condition, the mean performance of participants were slightly better when they used their oral skills, their auditory skills, to learn the words than when they used their reading/writing skills. This could be due to the perceptual association that exists between the olfactory system and the auditory system, where auditory sensory information reaches the olfactory tubercle through the hippocampus, processing the auditory information. Now my partner will go into the unique parts of our study.
So if our study is actually taken to a higher extent and actually done again, it could actually have a lot of benefits because as of now in the United States, 2.4 million students are actually diagnosed with cognitive disorders. And these students have learning disabilities that tend to interfere with academic excellence and personal growth and employment and language acquisition. And if our experiment is repeated again and we find out that if essential oils do actually help someone to learn a language, they can help these people who have difficulties with learning a language. And as we all know, learning language is associated with many benefits, such as job employment or exceeding in an academic environment. So this would be very helpful for those who are having a hard time with learning a language. So now Miriam will be going over the unique and novelty of our experiment.
Our study was unique because no other study has tested the effects of citrus essential oils on ability to learn a new language. And in previous studies, the two different paradigms, auditory and reading/writing, were not used before. Also, in our study, we had tested adolescents who are in their critical period to learn a new language. This concludes our presentation, and thanks for listening. We really appreciate your time.
Have you ever opened up a website just to find ads for the items you just looked up on Amazon? Or have you ever wondered why Google is able to predict exactly what you searched up? Do you know why this might happen? Well, it's because of search engine algorithms, which I, Type Dvorchin, and my partner, Jenna John, investigated in our study. As a background, digital literacy is the ability to engage with the internet effectively. This includes knowing about algorithm skews and search history tracking, both of which can cause what's known as the filter bubble. Because of this filter bubble, you might not get a full picture of the information available on the internet. In our study, we wanted to see if this can be taught through an educational video to college students. Students participated in a survey where they were either exposed to a control video or a video that talked about algorithms.
We hypothesized that using educational media like short videos would effectively increase participants' understanding of algorithms and tracking. After participants watched the video, they answered 2 free response questions about how the internet works. We then analyzed these questions and found there was a statistically significant relationship present between the variables of participants who would watch the algorithm video and participants who had referenced tracking in their answers, reflecting an increased understanding of digital literacy topics such as internet tracking and algorithms. So, why is this important? This is important because digital literacy is still being debated as an educational topic, because the tenets of digital literacy are consistently changing, which is especially evident in the current generation who are digital natives, meaning they grew up with the internet, and it becomes difficult to define what needs to be taught and what doesn't need to be taught. Additionally, algorithms shape a large part of the internet today, such as social trends and your tracking history, etc., etc. So, it's been agreed by social scientists and even policymakers that algorithms need to be made more transparent to the general public. Therefore, educational videos provide a good starting point for more formalized education for digital literacy in more niche topics such as algorithms and internet tracking to begin. Thank you. I hope this video was informational and gave you a brief insight on what our project was like.
Hi, I'm Sangeeta Chakraborty. I am a senior at the Bronx High School of Science. Right now, I want you to imagine that we're going out for a walk. Seconds before we turn the corner, we have very little idea of who or what we might see, but we still recognize quickly. This is an example of positional uncertainty, which occurs when a stimulus appears at a location randomly from a large set of potential locations. Uncertainty is common in our daily lives, and our perceptual systems need to cope with this in order for us to recognize things. To discover how uncertainty actually affects us, we measured its effects on visual recognition by the human observers and the ideal observer, or the optimal algorithm. The experiment was run on MATLAB, which measured the minimum contrast energy needed to achieve 75% correct. This then helped to calculate the efficiency of identification and detection tasks with no and high uncertainty. The results show that increasing uncertainty improves the efficiency of human observers identifying and detecting. We also found that the efficiency for identification was higher than for detection. Together, these results illustrate that humans attain the highest efficiency for the hardest task, which is identifying one of the 9 letters in one of the 104 locations. But what does this all mean? This study indicates that our vision has evolved to be closest to the ideal when identifying many targets under high uncertainty. It also supports the hypothesis that we're intrinsically uncertain. Multiple studies have been done that show the neuroscience behind our visual cortex, but we still lack this understanding of how the brain assembles neural data to make a recognition decision. But that's why this study is important for you to know about. We present the first measurements of how uncertainty affects human efficiency for identification and detection. Thank you. I hope you enjoyed the presentation. Stay safe and healthy.
Hello, my name is Theodora, and I conducted an analysis of short-period topological insulator superlattices, or TIs, through Fourier transform infrared spectroscopy, or FTIR. So essentially what a band gap is the amount of energy an electron needs to jump from the valence band to the conduction band where it can readily conduct electricity. So these band gaps are found in many materials, but the topic of focus here are these TIs. They're special because they can produce Majorana fermions, which allows them to be applicable to quantum computers and revolutionize technology as we know it. The issue is that they are susceptible to electronic surface defects, which can shadow their surface state properties. So to prevent this, the band gap must be engineered to become larger. So a common method of doing this is through growing superlattices in molecular beam epitaxy. So essentially what these superlattices are stacking semiconductor layers on top of one another, and typically on top of a sapphire substrate. We use bismuth selenide and antimony telluride in this case as they are well-researched TIs. So in the superlattices, quantum wells will form with adjacent minibands. So when these minibands thin out, a discrete band gap is formed and is believed to be larger in superlattices with thinner bulk periods. So what we did was we set out to prove that TIs should be grown on top of sapphire, that decreasing period thickness will enhance the band gap, and that stacking two TIs on top of each other will provide a more defined peak and corroborate initial band gap value. We collected all of this through FTIR, which produces an infrared spectra with absorption peaks that correspond to different frequencies of vibrations between bonds. So we collected an absorption spectra of sapphire substrate through FTIR, and the data we see shows absorption occurring in the 0 to 0.2 EV range. It flattens out and cuts off at 0.6 EV. This is excellent, as we're expecting to see peaks from 0.2 to 0.6 EV. So this proves that sapphire continues to be the correct substrate to grow these TIs on top of. So following this, we utilize the talc method to extrapolate the band gap. What this is just a linearization that correlates energy to absorption. A peak is expected where the jump from the valence band to the conduction band occurs, and we extrapolated band gaps from 3 different superlattices with period thicknesses ranging from 5 nanometers to 12 nanometers. So the trend that we were predicting rings true. The band gap does decrease as the period becomes thicker. So since these measurements were all conducted at room temperature, I wanted to solidify them more and suggested that we take 2 samples of the same superlattice and stack them for a more defined peak. So the graphs you see on the right show that the peaks occur in the same location or one of negligible distance. This corroborates the initial band gap values received. So sapphire proves to be an effective substrate to grow these TI superlattices on. If the period thins, the band gap will increase, and when 2 superlattices are stacked, more defined peaks are observed. We want to use low temperature FTIR for clear spectra in the future. Thank you.
Hi, today we're presenting our project that I've been working on since the 7th grade, and with my team through our high school years, we've drawn excellent conclusions.
Ever since we attended chemistry, we became very intrigued with the material we were learning. We found out that different combinations of salts and the spontaneous reactions from the voltaic cell we are able to produce energy. As you know, with a growing, evolving world, environmental problems such as climate change and global warming are inevitable. So through the generator, we want to produce a clean source of renewable energy for the future.
We have been researching our project well in advance of creating our own saltwater generator, and that meant that we looked up to other scientists who have created their own trial and errors to really give us an analysis of what this project was going to be about. And from all the articles that me and my group have read, we have concluded that raising the pH would also increase the amount of voltage produced. Diana?
To piggyback, with multiple test trials, we were able to collect data that was able to be processed into t-test and a bar graph. The t-test proved its valid realisticness, and a bar graph proved our hypothesis, and both of these helped to show our focus on the areas that needed most improvement.
To also piggyback of those areas that need improvement, we looked at the graph and we saw that the Himalayan and the rhubarb sea salt had the least amount of volts, and we questioned ourselves why. And we have found out that the rhubarb sea salt is like caviar. It's like a round cylinder ball. And from that, we can conclude that the more powdery salts produced more higher voltage. And then we pushed our questioning even more. Why are these powdery salts producing so much volts? And that is because of the higher surface area.
As for the further direction, we want to make sure that the generator is available to everybody. So we're currently researching to find cheaper alternatives to cut down the cost of the generator as much as possible. In addition, we are also trying to increase the scale of our project so that we could produce enough voltage to light up a light bulb or even charge a cell phone. Going back to the t-test, we want to make sure that our experiment was reliable so other scientists could corroborate for our experiment to come alive and make our big dream come true.
Along with our future directions and our future goals, we really want to make it able to go to the ultimatum and make it available everywhere around the world and probably change it while the chain reaction help out with global warming. Thank you.
Thank you.
Hello, my name is Hyun Kim.
And my name is Caitlin Wu.
In our project, we created a hydrophobic surface using electrodeposition with the precipitation of polymer chains.
Did you know that lotus plants are one of the most traditional plants in East Asia? It symbolizes both perfection and purity to the heart and mind. Not only is it very symbolic to East Asian cultures, but the microbumps and nanofibers of the leaves create extremely hydrophobic properties that pique interest in the material science world.
In our project, we developed a metal hydrophobic surface from the analysis of lotus leaf by using zinc electrodeposition with the addition of silicon layer. The electrodeposition mimicked the microbumps of lotus leaf, while the silicon layer mimicked the nanohairs. By measuring contact angle and sliding angle of our created surfaces, we were able to derive a relationship between contact angle and sliding angle. It was interesting because our background research stated that those two components are not necessarily related.
When looking at graph 1, we saw that after a very short electroplating time, 5 seconds to be exact, the contact angles of our zinc surfaces improved dramatically compared to when it was not plated. This verified that the electroplating method did in fact create hydrophobicity. The contact angles of the surface with silicon were also compared, but since the addition of silicon generally follow the same trend line, we concluded that silicon does not heavily affect the hydrophobicity in terms of contact angle. However, when looking at graph 2, we saw that the silicon did enhance the sliding angle, decreasing it to 15 degrees and then leveling out.
Graph 3 shows the contact angle measurement of copper. By collecting data for the contact angle of copper and seeing that the hydrophobicity was improved, it verified that the electroplating process can be done with many other metals.
You may be wondering, what makes this surface so important? It turns out that metal is used in everyday infrastructure such as bridges. As of right now, epoxy is used to prevent these surfaces from corroding away, but it fades and can create water pollution. With the development of a hydrophobic metal surface, bridges and other metals that contact water do not need to rely on an outside source of protection, and there can be a decrease in environmental pollution.
We hope that you enjoyed watching our video.
Thank you.
Hello, my name is Ethan Weisberg, and today I'll be presenting on my research conducted at the NYU Biomedical Chemistry Institute in the Dr. Kent Kirschenbaum lab. My research has been in the development of a biomimetic macrocycle cancer therapeutic. And so, this project started when I first identified cyclin-dependent kinases as a promising target for cancer therapy. In fact, they've been a promising target since the 1980s. However, the main approaches have been very flawed. Specifically, the main approach is using ATP-competitive compounds, which bind to the ATP binding site, disrupting CDK activity. This approach has two flaws. Firstly, it is not very specific to any CDK or to CDKs in the family of kinases more broadly. This can lead to cytotoxic effects. Additionally, ATP concentrations are very high within the cell, which means that it is very hard to develop a potent ATP competitive inhibitor. Thus, it is ideal to target protein-protein interactions. These protein-protein interactions have been explored for inhibitors. However, no drug-like inhibitors have been developed. The main inhibitors are biological peptides. Thus, what I've set out to do is to utilize the technology used in my lab, specifically peptoid macrocycles, as a potential inhibitor of this target that I identified. And so, what I did was I conducted a literature review to determine to look at structure-activity relationships with these biological peptides in order to design a peptoid macrocycle that could target the cyclin binding groove of CDK to cyclin A, the main target I was going for. This target had been previously shown to lead to tumor-specific E2F-induced apoptosis, so it was a good target for a cancer therapeutic. Additionally, peptoid macrocycles were a good material to carry out this goal because peptoid macrocycles have been shown to mimic peptide secondary structure, as well as being resistant to proteolytic cleavage and having enhanced cell permeability. So, they can mimic biological molecules well, but they have good pharmacokinetic properties. Thus, I went forward with synthesizing my peptoid that I designed based on this project I had come up with. I synthesized this peptoid using solid phase synthesis. I purified it using high precision liquid chromatography, reverse phase, and analyzed it using MALDI-TOF mass spectrometry, which revealed that I had synthesized the compound successfully with a 99% purity. In the future, I'll be analyzing its potency, and for now, while I'm at home, I'll be modeling it computationally.
My name is Ben Nicastro. My project deals with the process of sepsis, which essentially is the body's overzealous and harmful inflammatory response to foreign invaders and blunt trauma. Now, I don't have a lot of time, but suffice it to say, systemic inflammation and sepsis are one of the most, if not the single most pressing medical issue in the United States and the Western world. In terms of raw patient mortality, sepsis is the single highest contributor in the United States and in the vast majority of Western European countries. In fact, in the United States, sepsis accounts for 10% of annual deaths, period, which is a staggering figure. And individual instances of sepsis can be equally scary as their aggregate manifestation. The rate of mortality of sepsis is extremely high, especially among elderly populations. In fact, anywhere from 30 to 50% of instances of elderly severe sepsis result in death, meaning that for elderly patients, the chance of surviving sepsis can essentially come down to the flip of a coin. Now, for the past few decades, the Western medical world has pretty much had no biochemical means of dealing with sepsis. We've been focusing our efforts on the more on the fundamental source infection and instances of trauma that later lead to sepsis, which was, as far as I'm concerned, certainly not a misguided thing to be doing. But as our capacity to deal with the individual source infections increases over time, we need to be shifting our focus from figuring out ways to deal with infections and trauma to figuring out ways to deal with the results of those infections and trauma, which in many, many cases is sepsis. So that's what my project sought to do, and I won't go through all of the specific data, but essentially we discovered two novel peptides called M3 and LP17, which serve substantial inhibitory roles in the pro-inflammatory pathway mediated by TREM1, which is one of the largest pathways involved in sepsis. Not only in a binary fashion, but also in a dose-dependent fashion, meaning that as you increase the concentration of these peptides in cells that are stimulated, that are basically stimulated with sepsis, for lack of a more coherent explanation, the concentration of pro-inflammatory cytokines and chemokines decreases at a rate proportional to the increasing concentration of the peptides. In other words, there's an extremely high potential for these peptides to be used in a pharmaceutical and clinical role.
Hi, my name is Emma Yang, and my project is GADNet, a platform for connecting researchers via the genes and diseases they studied and will study, with the goal of expanding the world of genetics research. First, let me share with you the problem that we're seeking to solve. The problem is that there's an imbalanced research effort directed towards individual human genetics research. Current research is often guided by previous experimentation and previously understood genes, what we call the comfort zone of research. Most research currently only concentrates on approximately 2,000 of the 19,000 genes of the human genome. It also often focuses on the characteristics of genes, not their connection to diseases. There's also a lack of access to information between diseases and understudied genes. So the solution that we came up with is GADNet, a web-based platform that connects researchers and institutions via the genes and diseases that they study. It also helps researchers to explore understudied genes and to enhance opportunities for collaboration, to broaden the distribution of well-understood genes and discover lesser-known genes that could potentially be important, and that could help accelerate the progress of drug and target discovery. So now let me talk about kind of the process of developing this web app. There are 4 data sets that we use to source genetic and drug discovery research data. The first is PubMed, which provides published research papers on not only genetics research, but also the entire scientific field. We also use ArchS4, which is a massive database of publicly available information about the human genome, which includes data mined from PubMed. GeneShot provides data that links genes to other genes predicted to be similar to them, based on their structure and conclusions from published research. And finally, Diseases connects genes to diseases that have been linked to in genetics research. To develop the app, I used 4 programming languages. First, I used Python to pre-process a lot of the data that I got from the 4 databases. I then stored this data in a MySQL database so that it was easier for me to query and get the information that I needed for each search term. I then built a backend server using Node.js that interfaced with the database and the front end of the web app to get the information from the database. Finally, I used React.js to build the front end of the web app, including the search interface and the actual network that was generated each time the user puts in a new search term. The final product is a web app with a search engine, a visualization layer, and a natural language processing engine. The search engine is what interfaces with the SQL database to get the information that is necessary for each search term. For example, if I search for an investigator, it queries the SQL database for the genes that investigator has published about. Then for each of the genes that it finds, it queries the database again, as well as the GeneShot API to find other investigators working in the same gene, disease linked to that gene, and co-expressed predicted genes that are predicted to be similar to that gene. For diseases, it also goes to the SQL database to find genes related to that disease, and then finds related investigators, diseases, and also predicted genes. Finally, for a gene query, it looks for the investigators working on that gene, diseases linked to the gene, and predicted genes. All of this data is then sent to a visualization layer, which processes the data and makes it into a network, a visual color-coded network that allows the user to look at and process the data visually. So it's much easier for you to kind of look for the information that you need and to see what kind of connections are out there and opportunities are there for the genetic space. Finally, I also added a natural language processing engine, which allows the user to use an English-like natural question to look for information in the web app. For example, if I'm looking for a specific disease, I could say what genes are related to Lyme disease, or who is working on the BRCA1 gene. It's much easier and more accessible to access the information this way, and accessibility is a big goal of this project because all of this data is publicly available, but the main issue is that it's really difficult to actually go through that huge amount of data and to find the information that can help advance research or to help a patient better understand genetics research. Allowing us to present the data in this way, as well as allowing a user to put in a question, makes it a lot easier for you to look into that data. So here is a screenshot of the actual web app that I developed. We have an investigator query for a certain person, and you can see that you can have the person you're centered about in purple, and you have a list and a table of the genes that that person has researched and how many times they published for each gene, which are in light blue in the network. Then in yellow in the network, you can see related genes that are predicted to be similar to those genes. In red are other investigators working on those genes, and in white are the diseases that are related to those genes. And you can see how this could be useful for an investigator, because if I'm this investigator, I can see, well, this is the stuff I've been working on, and you can also see who else could I be collaborating with, or what diseases connections can I be looking into, or possibly what other genes can I start looking into as well in connection to my previous existing work. Using this, we can help to broaden the horizons of the genetics research and drug discovery in this way. Here you can see a really similar concept with Lyme disease. You can see the related genes, people who are working on those genes, and other diseases that are related to the same genes. And finally, here you have the natural language processing section, where I can ask, here I've asked a question, who is working on BRCA1? And I have a list of all the authors and also the network for the BRCA1 gene. So the next steps are to integrate additional data sets to expand the network scope. For example, allowing the user to search for a drug as well and look for information about that. I'm also looking into expanding the NLP capability so the user can query the database to ask more questions and have more powerful natural language processing to ask more complex questions, and also deploying the platform not only to researchers but also to patients to make this accessible and beneficial to people around the world who want to advance genetics research and want to expand this distribution of genetics research. In summary, GANA is a novel platform that has the potential to broaden genetics research, significantly accelerate drug discovery research, and to foster collaboration among researchers and institutions to develop drugs that could help benefit everyone. Thank you.
Hello. I'm sure that most of you watching this video have heard of genes, but have you ever heard about selfish genes? My name is Jaya Kim, and my research focuses on studying selfish genes in order to find new avenues of treatment for cancer. Selfish genes occur because not all genes in an organism have the same mode of inheritance. For example, in the case of the X and the Y chromosome, a female progeny will have 2 copies of the X, while male progeny will only have 1 copy of the X. A gene on the X chromosome can develop into a selfish gene, which tries to exploit this system by skewing the sex ratio of the progeny. This is selfish because if there are too many females, then the species' integrity could be compromised. However, all is not lost. This often then results in intergenomic conflict, where it is hypothesized that the selfish gene, also called a distorter, distorts the sex ratio to its advantage, while another gene, called a suppressor, acts as the antidote, suppressing the distorter and restoring the sex ratio to normal. I studied 2 intergenomic conflict systems that exist in the model organism Drosophilus simulans, a fruit fly. The Durham and Winter system, respectively, contain the distorters DOX and MDOX, and the suppressors NIMI and TIMI. However, there is currently no genetic evidence for this hypothesized system. My research aims to provide evidence for whether the distorters and suppressors are truly responsible for their hypothesized effects. I like to think of it as a detective story. Who is killing these fly sons? Here are our suspects, DOX and MDOX, the selfish genes. TIMI and NIMI are the vigilantes that are thought to fight DOX and MDOX. Through studying CRISPR null mutants, I could discover the roles of the different genes in intergenomic conflict. By seeing what goes wrong when the gene isn't there, I could find out the normal function of the gene when it is there. Through the first set of experiments, I found that TIMI and NIMI directly suppress the sex ratio distorter. Then, I also needed to prove that DOX and MDOX were the genes directly responsible for this sex ratio. Innocent until proven guilty, right? I then discovered not only that DOX and MDOX were responsible for the distortion, but also that they seemed to affect each other. I found that when I removed different combinations of the 4 genes I was studying, I got various degrees of sex ratio, suggesting that TIMI and NIMI seem to have synergistic effects in suppressing the selfish genes DOX and MDOX. This is significant because it is the first example of intergenomic conflict where this has been the case. My conclusions supported the hypotheses and established the genetic pathways involved in this intergenomic conflict system. We found the culprits and the heroes. It's a good detective story. These results are significant because knowing the genetic pathways involved makes it easier to fight cancer cells that mimic such pathways. Cancer cells are a selfish gene of a sort, and their genetics may not be the same, but it rhymes. With more knowledge of these intergenomic conflict pathways, you may be able to find a way to suppress the selfish cancer gene. Thank you.
I am Marisha Toa, and my project was on the effector retrotransposon 412 knockdown on locomotor function of Drosophila melanogaster. Aging is linked to a decline in health in all organisms, so it is important to understand genetic contributions to aging and find ways to prolong one's life and health. My lab looked at retrotransposons, which are genetic elements that replicate through an RNA intermediate, in other words, a copy-paste mechanism, and potentially causes DNA damage, which may contribute to aging. To study how retrotransposons affect aging, we used fruit flies and found that knocking down or reducing the expression of a specific retrotransposon, known as 412, extended lifespan in female but not male flies. However, increasing lifespan is not enough. To find therapeutic strategies against aging, increasing health span is also important. So how can we test health span? We can look at how knockdown affects aging phenotypes, such as the decline in motor activity. I hypothesized that knocking down 412 would reduce the accumulation of DNA damage and therefore improve motor function in older flies. To test the motor function, I tested the climbing ability of flies for 2 knockdown groups and 1 control group,
with 3 vials per group. By startling the flies down to a table, we induce a negative geotaxis response, which is the fly's innate response to climb to the top of a vial when startled. Then we counted the number of flies able to climb to each third of the vial. We record the data over the fly's lifespan to see whether the effects vary with age. In Figure 1, the percentage of flies at the top of the vial is indicated by yellow, middle by blue, and the bottom by green. Using a one-way ANOVA, there was no significant difference in the percentage of flies between the knockdown and control groups for the day 12 flies. In other words, despite reducing the expression, motor activity remained about the same. Why is this the case? Well, younger flies have a higher baseline stress resistance, allowing them to perform just as well. According to Figure 2, however, in older flies, knockdown groups demonstrated significantly better locomotor activity. Interestingly, knockdown also improved male locomotive function, although it did not improve male lifespan. Although it is unclear why, it is good that male flies could also benefit from reduced expression of 412. Based on the data, knockdown of 412 improved locomotor function in older flies as well as increasing lifespan. Although this study was conducted using genes in flies, it brings us a bit closer to understanding how to slow down the process of aging, even in humans. Future experiments will further help us understand how retrotransposons are involved in aging. Because many flies died throughout the experiment, future experiments should also use untested flies for each subsequent assay to maintain consistent N numbers. We can also test other aging-related phenotypes such as learning and memory, as well as starvation. I would like to thank my mentors, Dr. Julie Seacombe and Blair K. Schneider, for providing me with a great research opportunity, as well as my research teacher, Dr. Mahmoud, for his guidance and support in the classroom. Thank you.
Hi, my name is Shinyi Yang, and I'm a senior in the High School of Math, Science, and Engineering. Today, I'll be quickly discussing my project on a potential therapeutic drug on type 1 diabetes. Let's start with a quick history lesson. In January of 1922, Leonard Thompson was a 14-year-old boy dying from diabetes in a Toronto hospital. He became the first person to receive an injection of insulin. He died at 26. If this method almost doubled the life of a young boy, imagine what a potential cure could do for the 1.25 million Americans that are suffering from type 1 diabetes today. Now, synthetic insulin is only a temporary solution. It's costly and it is not a permanent cure. In comes dextran sulfate, a semi-synthetic polysaccharide, which previous experiments in my lab was found to decrease beta cell death, preserve beta cell mass, and restore insulin secretion. For my experiment, I treated insulinoma 1 cells, which are cancerous rat beta cells, with different doses of DS in the presence of cytokines to replicate in an inflammatory type 1 diabetes environment. I counted the number of live cells after overnight treatment, and using trypan blue to distinguish which cells were alive and which cells were dead, I calculated the viability of those cells. My results show that as the concentration of DS increased, the percent viability in the cells that were treated with cytokines increased as well. This shows that DS improves insulinoma 1 cell viability in the presence of cytotoxic cytokines. This is a dose-dependent effect, and the 2 highest doses of DS, 500 and 1000 micrograms per milliliter, provides the greatest percentage of cell viability. It is possible the higher doses of DS may cause cellular damage and death in beta cells, which requires further testing. My results can provide a baseline dose-response assessment for future in vitro and in vivo preclinical studies in rodent and/or human models of diabetes. If dextran sulfate were to be used in a clinical model, it would inevitably come in contact with immune cells, and so a future experiment could be to show how dextran sulfate affects the viability of immune cells like antigen-presenting cells. Hopefully, my experiment can prove as a stepping block for future experiments on dextran sulfate. Thank you for listening.
Hi, my name is Paige, and in my project, I was trying to learn more about how quorum sensing works in eukaryotes. So quorum sensing is the ability of cells to adjust their behavior and gene expression in response to the density of cells around them. And while it has been studied very extensively in bacteria, we have very little knowledge of how it works in eukaryotic organisms, despite the fact that studies have suggested it may play a role in tissue regeneration and cancer. In order to do this, I used a model organism called Dictyostelium discoideum, which has a simpler and easier to work with genome than human cells, but has highly conserved signaling pathways and has been shown to use quorum sensing to determine whether or not their cells will form fruiting bodies when starved, which is a process that's only beneficial at high cell densities.
So the protein in D. discoideum that I decided to look more closely at that's involved in their quorum sensing pathway is PKCA. One reason I was interested in PKCA is that it is an ortholog of human protein kinase C, which has been shown to be involved in development, cell differentiation, Alzheimer's, and cancer. The other reason is that we don't fully understand how it works in D. discoideum either, as both overexpressing and knocking out the PKCA gene inhibit development. The two main questions I had were what role does PKCA play in quorum sensing and development, and along with that, what proteins are acting downstream of PKCA in the quorum sensing pathway, so essentially how does it function in this pathway. To do this, I used a suppressor screen, which involved knocking out the PKCA gene, inserting random mutations into the genome, and then looking for a rescue of wild-type phenotype, in this case the formation of fruiting bodies after being starved for 24 to 48 hours. I was able to identify 5 cell lines with mutations that rescued wild-type phenotype, meaning that they did form fruiting bodies. And then by looking at how well they rescued the wild-type phenotype, so for example, how many spores were formed, how viable were these spores, and just the general morphology of the fruiting bodies, like how they were developing, what they looked like. I was able to piece together a model for how these 5 mutated genes might interact within the CMF quorum sensing pathway that is consistent with prior research showing that excess PKCA inhibits development and what I was sort of looking into in this project, why inhibiting it also inhibits development. Obviously, there's a lot more research that needs to be done. The next step is to identify which genes were mutated in each of these cell lines. And then once we do that, we can do more experiments that'll help either validate or modify the model that I have. And then finally, we can look for human orthologs of these genes, which could ultimately serve as targets for drugs designed to expedite the wound healing process or treat cancer.